Today I explain about the cell wall, their structure and dynamics. Cell wall was first observed and named simply a wall by Robert Hooke in 1665. In 1804, Carl Rudolphi and J.H.F. Link prove that cells have independent cell walls. A cell wall is a structural layer that surrounds some types of cells, situated outside the cell membrane. It can be tough, flexible and rigid, which provide. On the basis of chemical composition of cell wall, there are three types of cell wall. Bacterial cell wall, made up of mucopeptide and muramic acid. Cell wall of fungi, made up of chitin. Plant cell wall, made up of cellulose. Bacteria constitute a large domain of prokaryotic microorganism. The study of bacteria is known as bacteriology, a branch of microbiology. Bacteria was first discovered by Antony van Leeuwenhoek in 10,700. Peptidoglycan. Peptidoglycan, also known as murine, is a polymer consisting of sugars and amino acids that forms a mesh-like layer outside the cell membrane of most bacteria, forming cell wall. The sugars component consist of alternating residues, linked N-acetylglucosamine and N-acetylmur acid. These subunits, which are related to glucose in their structure, are covalently joined to one another to form glycan chains. Attached to the N-acetylmuramic acid is a peptide chain of 3 to 5 amino acids. The peptide chain can be cross-linked to the peptide chain of another strand, forming the peptidoglycan. Peptidoglycan structure. Types of bacterial cell wall. On the based structure of the cell wall and to their response to stain, bacteria have been classified into two types. Gram-positive bacteria, gram-negative bacteria. Gram-positive cell wall, usually thick, homogeneous, composed mainly of peptidoglycan. It accounts 50–90% of the dry weight of the cell wall. Contain large amount of teichoic acids. Gram-negative cell wall, multi-layered and more complex than gram-positive cell walls. Peptidoglycan of gram-negative bacteria is thin, comprises only 10% or less of cell wall. Outer membrane lies outside the thin peptidoglycan layer. Most abundant protein is bronze lipoprotein. Functions of bacterial cell wall. Protect cells against osmotic shock and physical damage. Regulation of substance transport into and out of cells. Contain supplemental genetic information such as resistance to antibiotics, production of toxins and tolerance to toxic environment. Take part in protein synthesis. Movement of cells. Fungi. Fungi are eukaryotes. Nearly all multicellular. Spore-bearing protists that lack chlorophyll. Two types. First is yeasts. Second is molds filamentous end. Multicellular. Fungal cell wall. The cell wall is made up of 1. Chitin. 2. Glucans. 3. Proteins. Glucan and chitin are components of the primary wall. Proteins are components of the secondary wall. Basic component of fungal cell wall. Secondary wall. Primary wall. Function of fungal cell wall. Protects against osmotic lysis. Acts as a molecular sieve. Contains pigments for protection. Binding sites for enzymes. The outermost surface of the cell wall. Provides a medium between the cell and the environment. A site where antigen and agglutinin gets attached to the substrate, host and other cells. Plant cell wall. The plant cell wall is a remarkable structure. Provides the most significant difference between plant cells and other eukaryotic cells. The wall is rigid, up to many micrometers in thickness, and gives plant cells a very defined shape. While most cells have outer membrane, none is comparable in strength to the plant cell wall. The cell wall is the reason for the difference between plant and animal cell functions. Because the plant has evolved this rigid structure. Components of plant cell wall composed of the middle lamella, the primary cell wall, the secondary cell wall, the tertiary cell wall. Plant cell wall structures. Middle lamella. It is present between two adjacent cells. It is situated outside primary cell wall and is made up of calcium and magnesium pectate. It acts as cement which holds the adjacent cells together. Primary cell wall. It is formed after the middle lamella. A thin, flexible and extensible layer. It is capable of growth and expansion. The backbone of primary cell wall is formed by the cellulose. The matrix is composed of hemicellulose, pectin compound, liquid and structure proteins. Structure of primary wall. Secondary cell wall. It is extremely rigid and provides strength. It is not found in all cell types. It consists of 3 layers known as S1, S2A, S3. It is composed of cellulose, hemicellulose and lignin. Structure of secondary cell wall. Tertiary cell wall. Tertiary cell wall is deposited in few cells. It is considered to be dry residue of protoplast. Besides cellulose and hemicellulose, xylem is also present. Plasmodesmata. Plasmodesmata are protoplasmic strands that connect the protoplasts of plasmodesma neighboring cells. Diameter is 40 to 50 nanometers. Composition of cell wall. The cell wall is mainly composed of carbohydrate material. The major components of cell wall are cellulose, pectins, hemicelluloses, proteins and phenolics. 1. Cellulose. It provides shape and strength to the cell wall. It composes 20 to 30% of the dry weight of primary wall and accounts 40–90% of the dry weight of secondary wall. 2. Pectins. They are a group of polysaccharides, which are rich in galacturonic acid, rhamnose, arabinose and galactose. Pectin are present in high concentration in the middle lamella where they presumably serve the function of cementing adjacent cells together. 3. Hemicelluloses. These are matrix polysaccharides built up of a variety of different sugars. They differ in different species and in different cell types. Xylem. It typically makes up roughly 5% of primary cell wall and 20% of secondary cell wall in dicots. This hemicellulosic polysaccharide linked with xylose and arabinose. 4. Proteins. Different varieties of protein are present in the cell wall, most of which are linked with carbohydrate-forming glycoprotein. The cell wall glycoprotein extensin contains an unusual amino acid, hydroxyproline, which is generally absent from the protoplast. Extensins are present in the primary cell walls of dicots, making 1 to 10% of the wall. Functions of cell wall. They determine the morphology, growth and development of plant cells. They protect the protoplasm from invasion by viral, bacterial and fungal pathogens. They are rigid structures and thus help the plant in withstanding the gravitational forces. They are involved in the transport of materials and metabolites into and out of cell. They are involved in the transport of materials and metabolites into and out of cell. They withstand the turgor pressure which develops within the cells due to high osmotic pressure. Thanks for watching.